Hello?
Yeah, go ahead.
Pause for a second. Go ahead.
Yeah. I would give you a very broad idea because I wouldn't want to go into the nitty-gritties. It would be like, say, it's a situation where you are using, say, an Ayurvedic drug in a medical condition. And you're trying to find out whether the medical condition is going to alter with the microbiome in that particular situation getting altered as compared to the non-disease condition.
I think
a broad kind of a situation, I would say.
So Mira, I think what Dr. Mira Nair has mentioned, I think that's a great approach, and probably Morris has given you the kind of the template how to do the metagenomics. So my thing would be killing two birds with a single stone. Are you willing to listen? So you're collecting materials for doing metagenome analysis, right? So it contains two different things. Once it contains the gut microflora, and it also contains the host material in it. Yeah. So you utilize the host material to do transcriptomics wherever possible. And you can use the gut microbe, the process to look at gut microbe. So you'll be able to get a much better understanding of the drug-drug interactions, of the drug-host interactions, because you're likely to see the bacteria to be, or the microflora to be the confounders, rather than be the actual cause and effect. So we really need to be assuring, just because you give an Ayurvedic drug, it doesn't mean you're going to see a change in microflora. There may be other conditions too that may be affecting it. So, and you may come to a fairly erroneous conclusion by saying, oh yes, I gave this Ayurvedic preparation and I see a change in flora between the controls and the cases. But there may be something else that's going on. So in order to do that, it would be much nicer if you can go and take a look from the host perspective. What are some of the genes that are being activated? What are some of the genes that are being inactivated? Especially if you're looking at certain proteases that are being produced in the gut. Are there any transcriptome messages that are you seeing? So you can combine several strata of information in order to make a more meaningful conclusion, rather than just saying, yes, change in microflora or not, doesn't truly answer the question. Did I confuse you?
No, you didn't. But I think you have given me an insight as to what is to be looked at.
Okay, thank you. Yeah.
Just to add to that, as Dr. Bhushan said, then you can make associations and causal relationships with what you're seeing with the host function as well as the microbial functions. You can make some hypothesis on that too, and can develop on what you're trying to look. So now you are in the middle of the hay and looking for the needle. Once you find the needle with these interactions, you go ahead and sew with it. So you get more and more answers with that.
Well said, Mira. And I totally agree. Don't, just because you tend to see a change in microflora, don't stick to it. There are, in fact, some of the experiments that I have reviewed for federal agencies and state agencies, we do find is they come to some rather findings which kind of bothers me to a certain extent, because it's, when we take a metagenome sample, it's a cross-sectional sample, okay? And you're not really seeing what is the time series events that are taking place, especially when you're looking at the dynamics of a microflora in the gut. So that doesn't tell you the big picture. So in order to do this kind of a study, you need to take several samples over a period of time before you introduce the drug and after the drug has been fully excreted. You need to follow the entire pattern and have different levels of metagenome samples that you have taken. And more importantly, when we look at some of the journals which I review for, we are now making it a point that just by doing one single metagenome analysis is not going to make it into the journal. We like to see at least 3 different independent runs in order to see external validation and internal validation of data that comes and truly says, yes, I'm consistently seeing the emergence of this microflora, certain microflora exiting from the process. So we need to show this consistently, repeatedly taking 3 independent test samples done at 3 independent times. So that's where we call as the rigor. The rigor in metagenome is being seriously questioned across many of the top journals. And in order to get into a good journal, you need to start fulfilling these conditions to begin with.
Yeah, I think that is the point. I think for studies where the target is the detection, metagenomics will be fine, but it will be difficult for those studies to show the causality, to show that something is causing something else. That will require much more rigorous study designs. Just sequencing at one point doesn't really show you what things are doing. That doesn't show you what is causing what. I think in diagnostics, if we are trying to figure out what we are missing using the routine diagnostics, then metagenomics might be helpful because we might be able to detect something we're not thinking about. But if we just want to say that this is exactly causing the disease, that is much more than that. You have to need more designs, more robust designs.
Thank you very much for all your inputs, sir.
Hello.
Oscar, go ahead. Yeah.
Yeah, myself, Dr. Rajesh Farande. I'm working as an assistant. Whether this Shannon and Simpson index are similar with what we are seeing in a principal component analysis?
No, actually, Rajesh. So the Shannon and Simpson index tell you about the abundance and diversity within the samples. So if you take a sample, let's say I have a fecal sample, two fecal samples, A and B. And when I say Shannon index, I'm saying how many species are there in A and how well they are distributed. So I will have a Shannon index for A and Shannon index for B, right? So within the samples, we are looking at the diversity. But the principal component analysis is looking between the samples. With all this information of sequences or species abundance in between A and B, how well they are related, whether A is close to B or A is far apart from B in a 3-dimensional plot. Are you getting what I'm saying?
Yeah.
Yeah.
Thank you.
Mm-hmm.
Dr. Zende, may I ask you, what are your big plans for genomics and bioinformatics in our MAFSU, especially in SSPH in Bombay Veterinary College? What are your big plans going forward?
Sir, that is what, sir, we are planning. We are keeping this training programs, and also we are.
I can't hear you. Little louder.
Hello.
Come a little closer to the mic.
Hello. Sir, am I audible?
You're audible. Yeah, the volume is a little low, as Oscar mentioned. Yeah.
Sir, definitely, sir. In this project, we are planning to have this bioinformatics center, because as such in our university we don't have. One sec, sir. One sec. Hello.
Now we can hear you. Yeah.
Now am I audible?
Yeah.
So definitely, sir, in this project, we are planning to have this bioinformatics center in our university. Because as such, we are, as Dr. Kurkure sir also told me that in Nagpur they have a small, but it is non-functional. So we are trying to have this bioinformatics center at least in Bombay or Nagpur. So we are planning, sir, we have to develop some manpower first, because as such there is no such a training program in institute or some bioinformatics lab also. So our Honorable Vice Chancellor is also insisting us to do something through this project, so that we can develop our manpower also and we can have this bioinformatics lab in future. And that is why, sir, that is our main objective through this project. So previously also, sir, we have discussed regarding this, what are the facilities will be requiring, and at least to start with something. The sequencer also we are planning, sir. But let us see which agency they can just sponsor for purchasing the sequencing. So,
okay.
so that part definitely we are planning in future.
So the one person in my lab who has worked on several different platforms is Morris. He has used an iTorrent, he has used a Minisec, he has used an Minine. So Morris, what is your take? What is your favorite instrument so far?
So now the favorite instrument for anyone is Illumina. That's the thing.
Yeah,
but personally, I don't know. I tend to like Minine. It all depends on on why you want to sequence things. But if you want to invest on something that will answer different varieties of questions, then obviously the Illumina will be the best choice. Yeah. But for detecting things, for people who want to detect things, especially if you are into detecting things in the field, Minine has really a very big advantage over the others. I mean, Oxford Nanopolar, and especially Minine because of the science. Yeah.
That highlight use of metagenomics for study of emerging and re-emerging zoonotic diseases.
Yeah.
So I can give you one example for emerging zoonotic diseases. We are talking a lot about coronaviruses, coronavirus evolution, mutation, so and so. From 2017, from the bat species, especially from Southeast Asia or China, there were a lot of metagenomic studies which highlighted rapid evolution emergence of new coronaviruses inside. So they have started using this study to highlighting why, what is going on inside the wild type species, and whether there is a zoonotic spill or not, right? I'm just putting it there. That's an example of how we use metagenomic studies to predict or model emerging or re-emerging zoonotic diseases. There are several other applications to it. I just want to give this, and I will just let Morris and Dr. Bhushan talk more, but there are an array of applications you can use this platform to answer the questions.
Yeah, Mira said it really well. So when we look at some of these platforms that are currently available in the market, like Illumina, iTorrent, they come with specific applications that they can give it to you. If you're really looking for screening for COVID-19 or you're looking for a certain type of a virus, they can create an application for you. So those are some of the advantages we are seeing more and more is these applications, in a quick turnaround time, they can make it for you and you can start using it.
And also, based on the question was if we can use metagenomics for emerging and re-emerging diseases. In my opinion, if you want to study a particular disease. You know the disease you want to study. I don't think in that case metagenomics is the best approach. Metagenomics would be a better approach if you want to study something. I would call it rather a kind of surveillance. So if you're looking at an emerging disease that you know, the best would be to isolate, to try to isolate. But if you want to find something that people may have missed, then you can use metagenomics, but that will be much more in terms of surveillance. That means you may run several runs, you don't find anything. So you would have to do it continuously over a long period of time to be able to detect things over time. But if you want to learn fast, or you want to to study, to investigate something which you already know, the best way would be to try and isolate that before you sequence.
Yep. So, when we are looking at when you're looking at zoonotic diseases or emerging diseases, unknown diseases, we have started working on a technique called a sequence-independent approach, where you can utilize that method where you can amplify all of the DNA fragments using Kenlo fragment 6 or Kenlo fragment 8, and then going in-depth sequencing of the amplified fragments. So we find that's one of the promising tools that we have, and we are currently initiating that big project. So hopefully in a year's time, again, keeping fingers crossed if all goes well, we should have this new type of surveillance tool. It's a surveillance tool, as Morris mentioned. You can apply this you can apply this on a nanopore-based technology. So we are really looking at developing CISPA-based technologies that will work, and then apply it for other platforms that are currently available, especially taking it to a point in place. If there is an outbreak, if we can do the extractions right away and put it on the system and start screening, because it starts screening in real time. So as soon as the data is coming out from the from the MinION, it gets analyzed. And if you set in target and say, look for avian influenza, look for African swine fever, look for brucella, it keeps looking for those signatures. And as long as it picks up a signature, it will stop sequencing there and highlight you and say, we have found something for you, which you had asked us to look for. So those are some of the highlights, and then you can overwrite it and ask it to continue sequencing. Okay, so that's the advantage we see, especially with the nanopore technology coming in to help us take it to what we use the word pen site assays, or pen site test, or cow site test, or farm site tests. These are the ones where we think is the biggest market. We expect the market to be close to around $20 billion in the next 10 years. And if we don't catch on the train and sit on the bus right now, we'll be left behind. So I would highly encourage every one of us to look into some of the nucleic acid-based methodologies that are coming in, because they provide significant advantages in making decisions for us as it relates to emerging infectious diseases. Another great thing that we as veterinarians and diagnosticians tend to do is we'll do a fantastic job of sequencing and producing data. The most important part is, how do you take it and convince that to your client? Your client is a veterinarian. He sends you a sample. You do all the sequencing analysis. How do you convince him in 3 minutes' time that this is the data that you're looking for, and this is how we can use it in our practice? That's the most important thing, and that's a big divide even today. That's the biggest divide, and we are making some serious efforts to bridge that gap. But finally, that's where we will start seeing more and more people start using genomics and bioinformatics, as long as we are able to convince them the data that we provide is valuable in their practice. So I think that's the biggest hurdle that we will face in the coming years of time, and we need to start addressing it right now.
Yeah. Yes, sir.
Dr. Bhushan, I think some of the questions as expected they are pouring in.
Okay.
May I go forward?
Please, it's all yours.
Yeah. Dhruva Desai, he wants to know if climatic difference makes any significance, then are we going to go for study of the Antarctic animal metavirome or microbiome? Yeah.
The more yeah.
Go ahead. Yeah.
People are thinking in the same direction as you, Dhruv. There are some studies which they have done looking at Antarctic marine microbiome or viral, and they also some studies on penguins and stuff. So look through the literature. There are interesting insights into it.
Dr. Morris, would you like to add?
Yeah, and I think all these studies. I think the next question is from that question to the next, the prospects of using metagenomics in rumen microbial studies. All these things depend on interest of people. Like, what exactly do you want to answer? Why would you want to do that? Especially one of the challenges that we've been looking at since yesterday is that these things are not cheap. These things are expensive. And then the question would be, is your question interesting enough to attract funders to give you money to do a particular study? Yes, using metagenomics, you can do a lot of things on rumen microbes. Same thing as you can use it to look at the viruses in Antarctica or any other region of the world. But that depends on how you present your question and what is going to solve. And then you get money to do the work, because I'm trying to imagine if you want to do the metavirome study in Antarctica. I'm trying to think of how much money that would cost and where would you get that money from. That would require to really show what the problem is and what is going to solve in the near future.
With regard to the question on rumen microbiome, this is Dr. Shannon. So Dr. Shannon, the person I would always refer to when it comes to the rumen microbiome and the work she has done is just phenomenally good. Her name is Dr. Deepthi Pita. She is from the University of Pennsylvania at New Bolton Center. So I would look up and read her work. She has done some excellent work as it relates to rumen microbiomes, and more so with relation to the methane producing type of bacteria and what some of the strategies that need to be adopted in order to reduce the innate expression of methane in some of these bacteria by modifying the diet. So she has some really good initial success, and she is one of our collaborators here at Penn State too, but she's at the University of Pennsylvania.
Okay. I think Dr. Vassar, some questions are there in chat box. You can just refer or we can.
I think so, because we are already referred to. Both the questions have answered.
Yeah. So can we?
As Dr. Bhushan said.
Sir, definitely, thank you so much. And Dr. Morris, Dr. Mira, and sir, your help will be very, very essential for us. And definitely, sir, we look forward to establish at least a small, minimal, basic bioinformatics lab at our university. And definitely, our Honorable Vice Chancellor and Director of Research, they are positive for developing such a kind of labs, at least in our university level, so that our postgraduate and PhD students, they will get benefited for their doing good quality research work, and they can be presentable at global level. So, before, sir, you are also visiting India, I think in the month of May. So before that, we will have 1 or 2 times meeting with our authorities, and we can plan and for establishing such a good quality lab at our university. Definitely, as we also signed the MOU with your university, and definitely we can go ahead for establishing such kind of the lab in our university level.
Yeah,
So, yeah, yes, sir.
We will look forward to that. Ravinder, my suggestion would be is to for MOFSU is to identify some of your faculty members who are going to be vested not for a project, 1 or 2 projects, but this is going to be their kind of their recognition for what they will be doing, or their major interest is genomics and bioinformatics. If you can list some of them, and we hopefully, if all goes well, Morris should be back at Penn State, keeping fingers crossed. So when Morris comes back, and with Morris and Mira, we should be able to do something really nice for you. Either you guys can send us people here to do a training, or I would be more than happy to send Morris and Mira to spend some time with you guys there.
Okay.
So let's look at either way which works best for you. So that would kind of establish the continuity towards genomics and bioinformatics.
Yes, sir. Yes, sir. Yes, sir. Yeah. Yes, sir. Definitely we can do that.
Yeah. Either way or both ways. It can be both ways.
Yes.
Yeah.
Yes. Yes. Yes.
Okay. If there's nothing more, thank you.
So nothing more, I think so. Yeah. So thank you so much, sir, for a very nice interaction with all the participants, and I think they have clarified all their doubts. And I specially thank our today's speakers, Dr. Morris and Dr. Mira. Have very nicely demonstrated how practically we can use this bioinformatics in their future research, and the students, they are doing the research at the PG and also the PhD level. Really, it was very good, interactive and informative lectures. And our Guruji, Dr. Jana sir, has moderated nicely, sir.
Don't say that.
Yeah. And your suggestions and your all for every activities of Bombay College and particularly MOPSU, you are always ahead and you are helping ahead right from initiation of this project. You have helped me out in all the cases, and hope same things will continue, and definitely the university will be excelling in providing the education to the students of our university. And they will be definitely, sir, once that the COVID situations will be get improved, and definitely we can send our students and the faculties also for the training programs over there. So with this, also I thank Dr. Vatkar sir, being co-chairman since 4 days, and tomorrow is the last day of our training program. So he's still there, sir. And Dr. Savani, he is also the rapporteur for this program. And finally, all the participants of this training program for participating nicely and interacting.
Thank you so much.
I request you to, I have sent the invitation later, and you have to send me by today for your bank details, because this closing March end is very, very near. So within that, I have to complete all the formalities regarding that. Okay, please. Thank you. Thank you so much, one and all.
Thank you.
Thank you.
Yep. Bye.
Thank you. Thank you.
Keep in touch. Yeah. Bye. Bye.
Yeah.
Thank you, one and all. Thank you,
Bye.
Bhushan.
Thank you. Nice seeing
Thank
you.
you very much.
Yeah. Bye.
Yeah. Very soon we'll meet.
Yeah.
Sure.
Thank you.
Thank you.
In? Yeah.